So today we're going to be talking about prokaryotic replication. And so we know that DNA replicates semi-conservatively, and we're going to be using the rolling circle replication model for our demonstration. So beginning with initiation at the replication origin, the DNA initiator A protein binds to begin DNA unwinding and the creation of replication forks. So now that that's happened, helicase, which is an enzyme, will come in and begin to unwind the DNA strands, driving into the replication fork and breaking hydrogen bonds. So because there's tension created due to supercoiling ahead of the fork, gyrase, which is a type of topoisomerase, moves ahead and relieves this tension. So now single-stranded binding proteins will come in and they will protect the different nucleotides from rejoining and forming a hairpin, which would disrupt and discomplete proper DNA replication. So now we're going to move on to elongation. And as you can see, we have the replication direction heading in the right and our leading strand on top with our lagging strand on the bottom. So now that we've moved on to elongation, primase, which is a type of RNA polymerase, will lay down RNA primers on the lagging discontinuous replication strand so that DNA polymerase will have something to attach to. So then DNA polymerase 3 comes in and begins at the 3' free ends of both the leading and lagging strands, adding nucleotide base sequences complementary to the coding and template strands. So at this point, DNA polymerase 1 will then come in, removing RNA primers and replacing them with DNA sequences, catalyzing phosphodiester bond formations. So nicks are left behind by DNA polymerase 1 performing its function, and between the Okazaki fragments that you can see, and which ligase, which is another enzyme, will then come in and seal these together, creating a complete complementary DNA strand. So as you can see, termination with prokaryotic replication is very different than that of eukaryotic. So as this diagram shows, rolling circle replication is designed to repeat many different times over, resulting in many different double-stranded circular molecules. So termination with prokaryotes are very different than eukaryotes, which will perform termination with telomerase and capping the end of the chromosomes and finishing replication that way. So to answer the question of what would happen if DNA polymerase did not perform its exonuclease activity in the 5' to 3' direction, it would mean that the DNA polymerase 1 did not remove the RNA primers from the sequence. Therefore, it was not replaced with any complementary DNA, which cannot be filled with ligase to perform a complete complementary strand. This means that DNA replication would not finish completely or properly and could result in serious mutations in the future.